In the previous class, we have studied the types of the tissue system on the basis of origin and the components which are present in it. But now we are going to study the types of the tissue on the basis of structure and position, that is location. So we are going to start with the tissue system on the basis of structure and location. The three types of the tissue system is distinguished in the plot. The first category, that is epidermal tissue system. Epidermal tissue system. It forms the outermost covering of any organ. Outermost covering of plant organs. It forms outermost covering of plant organs. It consists of two components. Yes, it comprised of two components. One is epidermis and epidermal appendages. Right? Now the another component is vascular tissue system. Now the vascular tissue system, it is present in the center. That was the outermost covering. It is present in the central part. It consists of complex tissue. Complex tissue that we have just studied now, that is xylem and phloem. Yes, xylem and phloem, and together they are going to constitute the vascular bundles, right? Except the epidermis and vascular tissue, rest of the tissue, it is going to represent ground tissue system. Ground tissue system. It is going to make the part of the plant except epidermis and vascular tissue. Now the ground tissue, it consists of three parts, that is cortex, pericycle and pith. The cortex can be further distinguished into three parts: hypodermis, that is just below the epidermis, then general cortex and endodermis. Got this? These are the different types of the tissue system. Now here in this lecture, I will be dealing with epidermal tissue system and vascular tissue system. And the ground tissue system I will deal when we will study transverse sections of different plant organs. Got it? So here I will be dealing epidermal and vascular tissue system. Ground tissue system I will be dealing when we will study transverse section of plant organ. In the epidermal tissue system, let's start first with the epidermis. Epidermis, outermost layer of any organ, outermost covering of any organ, surely going to be protective in nature. Epidermis consists of living cells, which are parenchymatous. Epidermis of the aerial parts, that is the parts which are above the ground, they are covered by a waxy covering, a waxy layer which is known as cuticle. So, stem, leaves, that is the aerial parts of the plant, they will have a waxy coating. That waxy coating is very much known to you, very well known to you, that is cuticle. Now, how this cuticle is formed? Because the epidermal cells, they show the secretion of a fatty substance, that is cutin. It is formed due to secretion of fatty substance by epidermal cells, that is cutic, right? Now generally the epidermis is single layered. Epidermis, generally it is single layered. But in some of the cases, the epidermis can be more than one layer, multi-layered. The multi-layered epidermis is seen in leaves of Nerium. The common name is oleander, and banyan tree, the scientific name is ficus. Nerium and ficus, both they are xerophytic plants. So xerophytic plants, so it means they are present, they are surviving in the water stress condition. So now the major water loss, it occurs through the leaves. So to prevent the water loss from the leaves, the epidermis is multi-layered. Got it? तो water loss को prevent करने के लिए through the leaves in these xerophytic plants, the epidermis is multi-layered. Along with that, multi-layered epidermis is also seen in roots of orchids. Now this multi-layered epidermis in the orchids, this is going to represent velamen. And what is its function? Now its function is absorption of water. Yes. No, it is not involved in preventing loss of water. It will absorb the water from the atmosphere because it is the part of the aerial roots. Right? Now, over the epidermis, over the aerial parts, in the epidermis, there is presence of the pores. These pores, they are known as stomata. Aerial parts, they have pores known as stomata. Singular is stoma. Stomatal apparatus, it consists of three components. Three components are there in stomatal apparatus. That is guard cells, GUARD, guard cells, then subsidiary cells, and stomatal pore or stomatal aperture. So now I will be discussing the structure of stomata in the case of the dicots and monocots. In the dicots, these are guard cells. Yes, guard cell. You write them as GC. So you can say the guard cells. See the guard cells, they are kidney shaped. They are bean shaped. The guard cells, they are modified epidermal cells only. These are all the part of the epidermis. Now epidermal cells, they are non-green, but only cell of the epidermis which is green, which is going to possess the chloroplast, that is guard cells. So guard cells, they are specialized epidermal cells which are green. They are green, they are going to possess chloroplast. Right? Now the wall of the guard cell, the cell wall of the guard cell is differentially thickened. Some part of the wall is thick, some part of the wall is thin. Let's see here. These walls, yes, these walls, that is the outer walls. The outer walls, they are thin. Inner walls, yes, these are the inner walls. The inner walls, they are thick. So outer wall of the guard cells thin, but the inner wall of the guard cells, they are thick. Inner wall is thick, but mind it, this inner wall is elastic in nature. It is flexible in nature. Around the guard cells, now there is presence of subsidiary cells. Around the guard cell, the cells which I am forming here, they are subsidiary cells. Write them as SC. The cytoplasm of the subsidiary cell and the cytoplasm of the guard cells, it is interconnected with the help of a pore. And you know that pore is plasmodesmata. Through the plasmodesmata, the two cytoplasm of the cells are linked. Now, in the center, there is presence of stomatal aperture. This is stomatal aperture. So the three components of the stomata in the dicots is very much clear to all of you. What is this? Guard cell. These are subsidiary cells, and in the center there is presence of stomatal aperture. Now coming to the monocots. In the monocots, the guard cells, they are dumbbell shaped. So the guard cells, they are dumbbell shaped. Again, these are specialized epidermal cells. Going to be here the chloroplast. Wall is differentially thickened. The outer walls, yes, these are the outer walls, they are thin. These are the outer walls, they are thin, and the middle walls, yes, these are the middle walls, they are thicker. Outer wall, thin, and middle wall thick. Now surrounding the guard cells, again there is presence of subsidiary cells. Again Surrounding the guard cell are the subsidiary cells. These are the subsidiary cells. And in the center, there is presence of stomatal aperture. So, what is the function of the stomata? Stomata, one, it is involved in the transpiration, that is loss of water in the form of the water vapors. Second, it is going to be involved in the gaseous exchange. So, the two roles, stomata, two functions, stomata will play. One, transpiration. Second, gaseous exchange. Right? Now after this, now we are going to deal with the epidermal appendages. Now the two kinds of epidermal appendages I will be dealing here. That is trichomes and root hair. So let's start with the root hair first. Let's say this is the cell of epidermis. This is a cell of epidermis, epidermal cells. From this epidermal cell, a tubular outgrowth will arise. A tubular outgrowth will arise. This tubular outgrowth arising from epidermal cell represents root hair. Getting it? So they are tubular outgrowth of epidermal cells. Now let's see what is there in the trichomes. They are not the outgrowths now. Let's say this is epidermal cell. Now they are not the outgrowth of the epidermal cells. They are extra cells present over epidermis. Right? So now they are not going to represent outgrowth of epidermal cell. The trichomes, they are also going to be known as stem hair. Now, trichomes, generally they are multicellular, usually multicellular. Root hairs, always and always unicellular. Trichomes, they can be soft or they can be stiff. Soft or stiff. When they are stiff, when they are hard, when they are going to have the covering of calcium carbonate. Root hairs, they are always soft. Trichomes, they can be branched. Can be branched, but the root hairs always going to be unbranched. Right? Now the trichomes, over the trichome there is presence of a waxy covering. What is that? Cuticle. They are cutinized. Cuticle present. But waxy covering is absent on root hair because the cuticle is the feature of aerial part to prevent water loss. Now trichomes, what is their function? They prevent water loss from the plant. And root hairs, they are involved in water as well as mineral absorption. The last thing which I can add here, that the trichomes, they can also be secretory in nature. Right? So after the epidermal tissue system, now I will be dealing with the vascular one. So now in the vascular tissue, I will be dealing with types of vascular bundle. So three categories I will be dealing here. Radial vascular bundle, conjoint vascular bundle, and concentric vascular bundles. Starting with the radial one. In the radial vascular bundle, the xylem, in this type of the condition, xylem and phloem tissue, they are present separately. Xylem and phloem tissue. Write X for xylem, P for phloem. They are present separately. It means they form different vascular bundles. They form different vascular bundles. They are present at different radii and alternate with each other. Present in different radii and alternate with each other. Yes? So here the xylem and phloem, they are not joined together. They will be forming different vascular bundles. This type of the condition is seen in roots. Roots of both dicot as well as monocot. So let's view its diagrammatic. Let's see its diagrammatic view. Right? So let's say this is the transverse section of the root. Here I will show separate xylem and phloem bundles. So this pink one, they are representing xylem strands. These yellow one, they are representing phloem strand. So you can see one xylem vascular bundle and one phloem vascular bundle. So xylem and phloem bundles, they are present separately. Xylem and phloem strands, they are present separately and they are alternating with each other. One xylem, one phloem. One xylem, one phloem. Now conjoint. As the name suggests, now the xylem and the phloem strands, they will join together. Xylem and phloem tissue join together, forming one vascular bundle. They are present together in one vascular bundle. Now, this type of the condition, it is seen in stem and leaves. In the stem, it is seen both in the dicot as well as monocot stem, as well as seen in the dicot and monocot leaves. Now, these conjoint vascular bundles, they are of two types. Let's see the first condition. This is xylem. This is phloem. So you can see phloem is present towards the outer side, xylem is present towards the inner side. This type of vascular bundles, they are known as collateral vascular bundles. outer side phloem, inner side xylem, one strand is present. If between the phloem and xylem there is presence of cambium, like this. Between the phloem and xylem there is presence of cambium. कैम्बियम प्रेजेंट है. अगर कैम्बियम प्रेजेंट है तो सेकेंडरी ग्रोथ के टाइम पर आउटर साइड पर बनेगा सेकेंडरी फ्लोएम, इनर साइड पर बनेगा सेकेंडरी जाइलम. दिस टाइप ऑफ द वैस्कुलर बंडल्स विच कैन शो सेकेंडरी ग्रोथ, दे आर नोन एज ओपन वैस्कुलर बंडल्स विच कैन शो secondary growth. These vascular bundles, they are known as open type, right? Yes. If the cambium is absent, secondary growth is absent, such type of the vascular bundle, now what do you call them as? Closed vascular bundles. Now, the open vascular bundles, they are present in dicot stem, and the closed vascular bundles, they are present in monocot stem. We have already discussed in the case of the monocot, cambium, collenchyma and phloem parenchyma, three of them, they are absent. Second category. In the second category, center, there is presence of xylem. Center, there is presence of xylem. Above the xylem, one cambium. Below the xylem, one cambium. Above the cambium, there is presence of phloem. Below the cambium, there is presence of phloem. So, xylem in the center. And on the two sides, it will have the patches of phloem as well as cambium. Such type of the vascular bundles, now they are known as bilateral vascular bundles. Center xylem, on the two sides phloem. So bicollector vascular bundle is seen in Cucurbitaceae, members of Solanaceae. Right? Now after the conjoined, now coming to the next category, that is concentric. In the concentric, one vascular tissue will form core and the other vascular tissue will completely surround it. One vascular tissue will form core and other vascular tissue will completely surround it. Two conditions can be seen. First condition, in the center there is presence of xylem. All around it is surrounded by phloem. Center में xylem, आसपास phloem. This type of the vascular bundles, they are known as amphicribal type. What is the another name for the xylem I have told you? Hadrome. And here the hadrome is present in center. That's why this vascular bundles, they are also known as heterocentric. Right. Second case, in the center now phloem, just reverse. All around it is surrounded by xylem. All around it is surrounded by xylem. This type of vascular bundle known as amphivasal. Right? So now in the center there is presence of phloem. The another name for phloem is leptome. So also known as leptocentric. Heterocentric vascular bundles seen in ferns. group of pteridophytes. Now the amphibians seen in Yucca. Got this? So this is all about the vascular tissue system. So in this class we studied phloem, complex tissue, then epidermal and vascular tissue system. In the next class I will be dealing with the sectional study of plant organs. Along with that also we will study secondary growth. Thank you so much.